if we think about what we just talked about when we left last time before the exam, right? I kicked the little cookie around and people got all grossed out that I ate it and it's still up there, so we'll have to go and check on it at some point. But we're going to continue on to talking about how we can understand what microbes need to grow, and then we can manipulate them because of media, right? So we start today with looking at the terms inoculum and culture. An inoculum is how we put organisms into a medium, right? A medium is their nutrition, where they grow, okay? And when we say cultures, we say we culture things. That means we put them on a media to grow them. But once they're growing on the media, we say that they are a culture. We do not say, right? We do not say, Isabella, pass me the infections, right? We say, hey, pass the cultures over, or let me have your culture of E. coli, or something like that. Okay, we don't say the things that are in the test tube are infections. Right? Although they can cause infections, they are not infections. Okay? So it's really important to understand those ideas, right? If we know what organisms need, we can design media to help us identify them. So if you remember from the first day of class, I said in lab that the functions of media were to grow, to isolate, to maintain, and to identify, right? That's what media allows us to do. And so here, if we understand where the organism is from, we might understand what it could be, and we can use special media to try to get them, to encourage them to grow so that we can identify them quickly. Okay?
Was grow, isolate, and identify?
Grow, isolate, maintain, and identify. Okay? Good?
Yes.
So, here we have some of the techniques we've already done before, right? This is a candle jar, right? And so the candle jar, remember, we can burn the candle. It increases the CO2 in the environment, and organisms that prefer CO2 are going to grow really well in it, right? We call those organisms that like CO2 what, Miss Amy?
Anaerobic.
No,
Anaerobic. Oh.
they're capnophiles. There you go, right? That capnophile. We call them a capnophile. Okay? This is an anaerobic jar, and this is a glove box. They're used for the same types of manipulations, right? We take away oxygen, and therefore we can grow organisms without oxygen. So organisms that will grow in there are going to be things like aerotolerant, anaerobic, or facultatively anaerobic, right? The anaerobic chambers that I'm used to working with are as big as this table. They're huge, right? And so we don't have those here because it's cost prohibitive. It's very expensive to run these things, and we only work very minimally with the anaerobes, right? And so we,
Anaerobic, air tolerant,
and facultatively anaerobic. Okay, good. So if we think about how those things might be incorporated into different ways of manipulating microorganisms, right? We can put the media into three big categories, right? The three big categories are chemically defined, artificial, and living. Chemically defined means that you have a recipe and that the media can be replicated anywhere in the world. Are you with me? So let's say Isabella's in Poland, and Amy, where do you want to go? Brazil. Amy's in Brazil, and I don't know. Where do you want to go, Samantha?
Greece.
Okay, Greece. So we got a person in Greece, we have a person in Poland, we have a person in Brazil. And if I am the principal investigator, I can send them everything, and I can say, make the media according to this, and it's all the same. We have a recipe, it can be replicated anywhere in the world. You with me? Okay? Chemically defined. If I then tell those same three people, go to the nearest meat market and I want you to buy 2 pounds of lean hamburger meat. I want you to take it back to the lab. I want you to boil it up for 4 hours. I want you to remove the supernate, the fat, and then I want you to use that broth as a media, right? Do we know exactly what's in it? No. We know that there's a lot of nutritional value in there. It's like soup, right? It's beef broth. But we don't know exactly what's in there because what the cattle might be consuming in Greece is going to be different than the cattle, whatever the cattle are consuming in Brazil is going to be different than what the cattle are consuming in the European Union. Are you with me? So all those things are going to be different, and so you are what you eat, and so are cattle, right? Most of the time they get fed either hay or cubes or whatever, right? Molasses or whatever. But in some cases, I don't know, maybe in Greece they serve them mojitos only, I don't know. And then in Brazil, pineapples, and in Poland, what's a famous fruit in Poland? Okay, fine. Alfalfa in Poland, right? And so those cattle are consuming that, they're storing different nutrients in their muscle tissue, and therefore the broth are going to be different, right? That's a natural medium. We don't know exactly what's in it, but we do know it's good. We know it's going to work, okay? And then living medium is something that is alive. Now we used to use living things all the time. We used to use rabbits and hamsters and all kinds of things. We don't do that very often anymore because we have all these molecular techniques, right? And we move from the organismal level to the molecular, the cellular level, to study how disease works, right? So I always tell people it's important to understand what we're doing, but it's really important to understand that a lot of disease happens at the cellular, at the molecular level, right? If something goes wrong with an enzyme, it's going to affect the cell, and eventually it's going to affect the tissue, and it's going to affect the organ, it's going to affect the organ system, and eventually the organism is affected, right? We do have living media that we use. We still use rabbits in some cases. We still use hamsters and guinea pigs and things like that, but it's rare. And some of those are only like to do safety tests to make sure that there's not going to be an adverse reaction in a patient when we're giving them this medication, right? A lot of times the animal survives, but that animal is a lab animal. It's a white organism. Eric, what would happen if we allowed white rabbits to run around the golf course out there? Well, no, they wouldn't proliferate. They're white.
Well, they might get massacred.
They're going to get eaten very quickly. There could be some hawks up there that are going yum yum, and they're going to catch them. Right? They will not survive. Okay? But the most important of all living things that we use as a medium these days are these beautiful cells that we call HeLa. Anybody know what HeLa is? What is HeLa? Christian? Oh, somebody else?
You said natural?
No, living. What's HeLa? Christian? Simple Agata. He said, I don't know. Who knows? L. Oh, Amy. Amy has an idea.
No.
Okay.
Exosomes.
HeLa is from a very famous cell line that was from a patient in the late 1950s called or named Henrietta Lacks. And so we got HeLa from the HE from Henrietta and the LA from Lacks. So it became HeLa. These are cancer cells, right? So in the 1950s, when they were trying to help this lady, they took some specimens from her tumor. It was so aggressive they couldn't do very much for her. But the physicians put all these cell lines, all these cells in deep freeze, and just forgot about them for a long time. It wasn't until the late 60s, early 70s that people started to look at these a little closer and found out that if they continuously fed these cells, they proliferated. They grew like crazy. And so usually science moves millimeters in advancement. But because of the onset of the use of HeLa cells, we moved a meter. There was just so much knowledge that we gained about pharmacokinetics, about pharmacodynamics, about cancers, about viruses, all kinds of different things because we could use those cells.
HeLa cells are cancer cells?
What's that?
HeLa cells are cancer cells?
Cervical cancer cells for
A
men.
specific woman.
A specific
They took
woman.
them from this woman. I think they still use them, right?
Oh yeah, we still have them. We have them here.
From that same woman?
They just
They just proliferate. Yeah, and we give them away. We don't sell them anymore. We just give them to each other. Yeah.
Oh.
I mean, you can buy them if you want to sell start your own cell line, but usually, if somebody calls up our biotech group, can we have some HeLa? Oh yeah, sure. Because we grow them so fast, we just give them away. Right? No, we're not gonna, if you call Isabel, can I have some Gila? And like, what are you doing? Oh, I just want some for my house. No. Okay? But, you know, we give away as research tools all the time.
When did that happen, and what year?
1957-ish, somewhere around there.
Oh, okay.
I read an article on that, the family was not happy with that.
The family did not know.
I was just gonna ask about that, like, did
How
her
does that
family,
work?
I feel like it's kind of, not an invasion if she donated them willingly.
She didn't donate them.
Oh.
But the physicians at the time did not know their value. They collected them to try to help her, and they just put them in storage to study later. They had no idea of profit or anything. And so this is a very famous case that has actually gone all the way up to the Supreme Court, right? Because one of the lawyers got involved and told the descendants of Henrietta Lacks, like 4 generations later, oh, you should be getting all this money from the biotech group because they're using your great aunt's cells to do all this research.
And they named it after her, which is kind of weird.
But the court ruled, as I would have hoped, is that these cells have been in the public domain so long that nobody's really profiting from them.
Yeah.
We've given them away to each other, that really they belong to the public.
And at
And
that
that's what,
point,
huh?
that at that point it was too far, like,
Yeah, who are you going to,
Compensate? Yeah.
who's gonna compensate?
Yeah, she-
And the lawyer said, well, J&J is a good example. I'm like, yeah, right. J&J's not gonna give you anything, right? But he named like 8 companies that have all
but
been
it's
using
wrong,
it.
that they just kinda took it and like didn't
But in the 1950s, there was no HIPAA. There was
Yeah, that's
nothing.
true.
There was nothing to protect the individual. And these people who collect died before they even started being used. They didn't appreciate it. I don't like that word because Jess doesn't like it. So they didn't appreciate any value from those cells. So the courts basically said, well, nobody's really profited from these things, so there really isn't any monetary value of them. The value is purely in research, and since they're in the public domain already and people are giving away, it's really hard to justify compensation for them.
That's crazy.
What's that?
That's so crazy.
Nowadays, you know, you gotta sign forms and all kinds of
There's
things,
a bunch of red tape.
but back then there wasn't. Yep.
Question about the media here, between the natural and the living, the natural, is it then just dead tissue?
Dead tissue. Well,
Well,
live tissue for just a little while until you start to cook it up or whatever.
processed.
Yeah.
But probably not easily replicable across the world.
Not at all, because they're so different no matter where you're at.
And the living used to be rabbits and hamsters, but now they're using what?
Cell lines.
Cell lines.
So not only do we have HeLa cell lines, but we have monkey kidney cell lines. We have rat lung tissue cell. I mean, we have all kinds of different.
Just
things.
like we hear this.
Yeah, because we've learned that if we just continuously feed these things, we can grow these things, and they can be of huge value to us. Think about it. We don't have to kill a lot of animals anymore for scientific
They're
discovery.
just keeping the cell lines going.
Just keep the cell lines growing. Yeah.
So could natural be like cow manure or something like that?
Yep. Exactly. It could be. But when we do use it to grow mushrooms on, how many people like mushrooms? They grow it on poo. So that little black stuff that you Samantha, you think I was kicking around poo? Do you eat mushrooms? Okay, good. You're eating poo too. Yeah. Okay. Fine. You can believe whatever you want, Isabella.
Can anybody like take their cervical cancer cells and like technically have them be HeLa cells? Or were the HeLa cells like super unique?
They were very aggressive and they grew very fast. So not all cervical cancer is fairly invasive, but I don't think anybody's going to be worried at the time they have cancer that maybe I can profit from this. I don't think they're going to be thinking like that, right? So, but yes, we could grow almost all cancers now. We could even grow blood tissue. We could grow anything you want, and if we have the right stem cell, we could encourage that stem cell to become anything we want it to become.
Looks like nowadays people can grow their ears and their arms to replace their.
Okay, yes,
replace
you could do that, but we're not going to go that far, okay? If you want a new ear, we can grow it on a plate.
Yeah, he like grew it on his arm and then they put it on his ear. It was weird. It was super weird.
So I don't know if you know this, but we are very closely related to the pig. And therefore, they've had the very early transplants for hearts were with hearts from pigs. And before, when we didn't have insulin that was coming from yeast, right? Because we've trained the yeast to do this, we would take the insulin from pig, and people who were diabetic would take insulin from pigs, and it worked the same way.
Right.
So, I mean, see how the progression of science has occurred over many, many years, right? And so we don't use, very rarely do we use insulin from pigs. We use insulin now that comes from genetically modified yeast. Yep.
Would any of the pig transplants, heart transplants take?
And theory and hypothesis, no, because they're marked differently than the
Yeah.
body. So the way they got around that is they give immunosuppressants to the individual, but that individual who received that pig heart knew that they probably weren't going to survive very long, but they were going to die then, and so they decided to donate their time and whatever was left of their bodies at the time to medical advancement. And I think one of them survived 6 months. Yeah. But I mean, complications and all kinds of stuff.
Yeah.
Because we were just learning about what can take.
Yeah.
Nowadays, we're so good at finding really nice matches for people who are going to get any kind of transplant. That still doesn't mean that your body's not going to reject those tissues, because every one of the cells in your body, with the exception of your white blood, your red blood cells, are all marked Isabella. And even if you and I were best friends and you gave me your lung, my body's going to say, oh no, this lung doesn't belong here, and they would attack it.
It only works with twins, right?
Not even then. I mean, even if you have that type of a isograft, the marks are going to be a little bit different. Much better chance coming from a family member, not even twins, but a family member, right? But there's still some chance of rejection.
Question, I just want to clarify that for the media for chemically defined, could we give us like an example could be just like
Peptone glucose acid agar.
okay.
Okay. Well, you asked for an example.
I was like, okay, I know fats and glucose. That's all like, okay. I know glucose is sugar, okay. So sugar, like agar, okay.
I
So
can
the
control
natural-
everything that goes in that medium.
Okay.
Okay.
And then for the natural, you said it just consumes like.
I'll give you an example of that one.
You said cattle.
But
something
you
like-
want an example?
Yes, please.
Brain heart infusion,
Okay.
also known as BHI media.
And
So
then
we
for the
take
living,
brains,
just-
we take hearts of animals, we put them together, we cut them up, we dry them out, we pulverize them into a powder, and then we mix them with DI water and sterilize them. Very good media.
Huh.
Good. I mean, all those parts are not usually consumed by Americans. Right? Oh, that's right. But we can send them across the sea. People will eat them out there, but not here. Although I kind of like the heart. It's good. But I won't eat brain anymore because of prions. I just don't. It's a delicacy. We call them what? Eating brain on a Sunday morning. Well, it's not barbacoa. Barbacoa is cheap meat.
I love
It's
barbacoa.
called sesos.
What?
You don't never had sesos?
That's not from my region.
What kind of Hispanic are you?
That's not from my region either.
Sesos are brain. You got them? Look at, she knows. They take the whole cattle, they put it in the ground, they cook it, the head, they pull it out, they crack the skull, you eat the brain, you suck the eyeball, it's all good.
Well, my
I don't know
grandma
what the eyeball.
Lightweight. But I don't eat brains anymore just because of prions. Just scary to me. Yep.
And so for the living, you just wanted to use the example. of
Hela
the
cells.
Yeah, the hema cells. Perfect.
Okay. You can say Amy's big toe if you want.
But prions hanging out just in the cattle's brains? Or are they everywhere?
Well, I think they're everywhere.
You said mainly deer.
Right now, we know that they're in deer in Texas.
Follow-up question on that.
Okay, hold on.
Same.
But we're not sure. We know, we check cattle if they act funny, And we know if we ever have bad, we have not had bad cattle in Texas yet, right? But deer came from another state and they brought them here to breed them, and they brought prions to Texas.
We live in South Korean stuff.
What's that?
The zombie deer, you know what I'm talking about?
Yes.
Is that the prions or is that
Yes.
the parasite?
No, that's prion. Don't call them zombie deer.
that's the term that people use when they see them.
I don't like that.
We've
Just-
seen one before.
Just call them wasting disease.
Yeah, that.
Why,
I couldn't
why
remember
is
that.
it zombie deer?
Because
Yeah.
they look.
Because they have no control over the
Yeah.
body, and when they walk,
They could have a completely like separate
have no
leg
control.
that's just
Oh.
chill, and they still act normal. Like not normal, but
So
like
it's
they don't have any sense to
they
us.
lose cognitive and neuromuscular ability.
Mm.
It's really creepy.
So if you ever see any animal that doesn't look like it knows what it's doing,
Alright.
stay the hell away from it. Call animal control. I don't know what that is, but something smells really good. Yep.
My neighbor is a deer hunter and he gave us some meat and he took it to a butcher shop, but do you think,
I would eat that meat.
okay, I know, and I have.
If you don't want it, you can give it to me.
No, it's from Johnson City, but I brought it up to my fiance. I was like trying to explain it, and then he's like, "Well, do we eat it or don't we?" And I was like,
If you don't want it, give it to me.
"No, I'm gonna eat it."
Okay, good.
But how do you tell? Is it just because
You
of
can't.
that zombie?
You
deer?
can't really tell. Don't call it zombies, okay?
Okay.
I'm sorry.
What if you eat a deer with frying prions?
You're gonna die at some point. But let me tell you
We're all
something.
gonna die
Even if you
at
cook
some
it-
point.
Let me tell you something. You're gonna die at some point. All right. Whether it's from prions or something else. I don't want you to not live your life because you're afraid of infectious agents. Does it look at me? Does it look like I've changed my life? No. So you all should not be afraid of what's out there. But it's good to understand what's out there.
Are the prions like mainly in the brain? And the meat is good. So if you eat a deer that's infested with
Well,
prions,
so
just don't eat the brain.
with deer, we know that the way that they get transferred from deer to deer is from saliva.
Mm.
So you know, when deer come up to each other, they don't shake hands. They rub on each other to say hello, right? Have you ever seen lions? They do the same thing. They rub on each other to say hello. Well, that's the way a lot of the the things that are in the kingdom of animals all like to be touched. And so a lot of them act like that. They're very social, right? So I just want you to understand what's there, but I don't want you to fear every single thing. Because then you just never leave a house. You become like my cousin. He won't leave his house. Oh no, there's too many things that can kill you. I'm like,
Is that because of you though?
no. He doesn't like me to come over because I'm not afraid of anything, and he's afraid of everything. And so I come in and I sneeze, and he's like, "That's it. I'm gonna have to like sanitize my tire."
So there was a type when we hunt any kind of animal, we always check for, I can't remember what it's called. I'm trying to. figure it
Tania,
out, the
probably
white.
Tania saginata. Mhm.
Yeah, it's the white that's on the meat.
Yeah, there's cysts of tapeworms.
Yeah.
Yeah.
And it just kind of looks like a film almost. I don't know. We always check for like they're like pockets.
Yeah.
on it.
Oh, in the meat.
And that's the only thing we've ever checked for. Like
That's more than most.
when you butcher, you just make sure that because we don't like to process our deer without processing it first ourselves, like taking the hide off because we don't.
We call that
trust
field
butchers.
dressing,
Yeah.
right? So
We don't trust any butcher.
So that's more than most people do. Most people don't shoot a deer and then they just take the whole deer to get processed. They don't even look at it.
Just make her bonus. He's too rich.
So,
He's like, I don't care.
It's stuff.
He just takes it. He's like, I don't want to deal with it.
any other questions about craziness? Okay. We can take these ideas and we can make and manipulate media. So an enriched medium is something that we've added something special to it to encourage the growth of certain organisms but inhibit others, right? So this is chocolate media. That does not mean it has Hershey's in it. What it means is that there's blood that has been cooked to the point where the blood is broken apart and it's turned brown and now it looks like chocolate. Heme has been released, okay? Certain organisms will only grow in the presence of heme. like Haemophilus influenzae and things like Neisseria, gonorrhea or Neisseria meningitidis. Okay? So these organisms will only grow in the presence of heat. So we can use this to really isolate them. Okay? Good? And it's called enriched media. Selective media is we've added something to the medium so that it inhibits certain groups of organisms, but it allows other groups to grow. So for instance, with PEA, phenol ethanol agar, we've added alcohol. And so if we think about the gram specificity of organisms, which gram specificity, gram positive or gram negative, are going to be more affected by alcohol?
Negative.
Gram negative, because of the outer membrane. So this particular medium inhibits gram negatives, but allows gram positives to grow. It doesn't kill gram negatives. The alcohol will eventually vaporize from the media, and then the gram negs will start to grow. But they slow them down enough of the time so that we can do whatever we have to do with the gram pos. Because the gram negs are more aggressive.
So it inhibits the growth of gram negative to allow gram positive.
That's
to grow.
PEA. The other way is deoxycholate. If you have an 8 at the end of anything, what does that mean, Clarissa? What does that mean? Clarissa is. Oh, she's over there. Okay,
But I
Clarissa.
don't know.
Oh, you don't know. Okay. What does that mean? Have an 8? So nitrate, citrate, phosphates, glutamates. What are those things? Salts. Salts. So if you have salts, which gram specificity is more susceptible to losing water because they're not protected for
Gram
osmotic,
positive.
gram positive or more likely to lose water. So deoxycholate allows gram negatives to grow, but it inhibits gram positives because they've all become dehydrated. Isn't that cool? And so if you think about that for just a minute, we sell a lot of things with a lot of salts in it, right? All deli meat's got a lot of salts in it, right? A lot of fish have brine, right? And the reason that those increase the salt concentration is to not allow for botulism to grow. That's a preservative. That's exactly right. Right? Good. But it works in the same idea. Yep.
How do you spell botulism?
Botulism.
DOCHLASM?
Yeah. Botulism. Okay. Good. Questions? We can take it a step further. Ellie, remind me. What are the purposes of media? What's the functionality of media? What does it do for us?
We use it to maintain. We use it to something with an I.
Identify.
Isolate, identify.
And grow.
And grow.
2 G's and 2 I's, right?
And an M.
Yeah, okay, good. Maintain. That's right. Okay. So if we think about this, we can do that with differential media. Now, we've been doing differential stains in the lab for the last 2 weeks. And differential stains do what for us, Jinjin?
Shows different morphologies and whether it's gram positive or gram negative.
Can show us
Or-
different types of cells or different parts of cells, right?
Yes, like endospores, for example.
Right. So differential media will allow us to differentiate different types of organisms based on actions that occur in the media. We've already used 2 differential media already in the lab. Can you name them, Tram? One of them.
The broth,
Which broth?
the thio.
The thioglycolate is it differentiates based on the stratification needing the need for oxygen. What's the other one?
What was that one called again? I forgot.
Thioglycolate. What's the other one?
Said motility.
Motility medium. It allows you to differentiate whether something is molar or non-molar. In this case, if we're looking, for instance, like salmonella, I can put it on one of these plates and it's going to turn a different color if it's salmonella because I'm targeting only that organism. Because I know what it produces as an end product of metabolism. And so therefore I can use that to my advantage. Okay? Good?
So the action that occurs on the media is mostly how it grows.
It can be how it grows. It can also be that we've added a pH indicator and it's changed color.
So you wanna, it can help in multiple, to identify multiple different.
So the definition of differential media is a medium that allows us to differentiate different types of organisms based on an action that occurs in the medium or a color change that is associated with a change in the medium. Okay? Good. You look very different in your picture on here. And therefore, I get you. Good. All right, so looky here. This is blood agar. Blood agar, we've added 5% sheet red blood cells. My question to you is, if you analyze this medium, look at it. Is it selective? Is it differential? Is it enriched? Or a combination thereof? Jess. Oh, okay. Isabella's. Jess, we'll give you first because I called your name first.
I first said enriched, but then I said maybe different, right?
Well, you hit the 2 on the head. It's both of those. It's enriched because we've added sheep blood cells to it. And it's differential because there's a change that's occurring in the media based on the toxins that are being produced by the organisms. The organism here produces toxins that destroy all blood. Isn't that scary? Right? And therefore, that's a really dangerous organism to us. That Streptococcus pyogenes. Questions?
All three are streptococcus.
No, there are 3 different organisms. That's Streptococcus pyogenes, that's Streptococcus pneumoniae, and that is the virulence group of Streptococci. They're 3 different organisms. Okay? How do you know that? Well, because of the way that they're acting on that medium.
This one is the most dangerous because why?
It is completely destroying all the blood. You see that? The second one is partially, you see a green zone around the colony? So it's dangerous, but not as dangerous. And the last one is not doing anything to the blood at all.
Okay.
Growing.
It's just growing on it, using whatever else is the nutrient source, but not the blood. Now, don't get me wrong. Anything that's in our blood is not good, right? But in this case, this one's not going to cause as much damage as the other 2. Yep.
So that wouldn't classify as differential, even though it's making like different.
It is differential.
It is differential, so it's all 3 of those.
No, it's not selective.
Oh. So enriched and differential?
Yep. But that's what Jess said earlier. Yep.
So you're saying that first one is more dangerous because it's technically eating that blood to grow?
Eating.
Not
How
eating,
about
but like consuming
digesting?
it. Yeah,
Okay.
yeah, okay.
Yes. Good. Good. Questions? Now, we can take all these ideas and put them into an understanding of how organisms grow in a population. Okay? So this is the standard growth curve. I've drawn my standard growth curve up here. And I'm going to remove kilo. But this is the standard growth curve, right? So the standard growth curve, in most cases, is only going to be described in 4 different phases. I don't like 4 phases, I like 5 because it's more descriptive. So my 4 phases, I've marked them. A is the lag phase. B, this one here, is the log phase. Log stands for logarithmic. Some people call it exponential growth. C is stationary. D is not normally talked about in most pieces of text or wherever else you might look. It's denouement. Denouement means rapidly declining. And then E is death. Although it's not really complete death, it's as close to death as a population can come. Yes, sir?
So that's your map for your growth curve?
Uh-huh.
Okay.
Correct.
Assuming D and E are on the right side of the.
Oh,
Yeah.
I somehow replaced it.
The y-axis is number of.
Number of microbes, microorganisms. Okay? Actually, that number. Because another way of saying that, Mr. Eric, is population.
What does C say, stationary?
Stationary. Okay? So, let's put it into a scenario. Samantha, open up that blind and look out. What you'll see is there's a pond out there by the golf course. The golf course, especially the older ones, are all built so that the holes all slant to a pond. Therefore, when it rains, everything washes down the fairway and into these ponds, and therefore the fairway is not moist and you can play golf when it rains. Yeah. Doesn't work out here anymore because lo and behold, the Riverside golf course is on a floodplain. And that's just the way it is. So it floods out there. If it ever rains a lot, you can go out there with a little paddleboat and go around there. But let's talk about this little pond, right? This little pond right now, if you think about it, it's dry, right? It hadn't rained in a while. It's dry. There's a little bit of moisture in there, but not very much. Is there anything alive in there right now? What's alive in there? Bacteria, probably, things that can survive with minimal moisture, minimal nutrients. Fair enough? Is there anything in there that's gone dormant?
Yeah.
What?
Fungi maybe?
Fungi maybe? What's the other?
Algae?
Algae? No, not algae.
Or plants?
Not plants.
Why not algae?
Maybe bacteria, maybe insects.
If they don't have water, they're not going to survive.
Archaea.
Huh?
Archaea?
The archaea maybe, but we're missing the most important part. Starts with an E, ends with an E. Has an N in it.
Endospores.
Endospore. So all the organisms that produce endospores have gone endosporic. They're in there because there's no nutrients or very few. They've gone dormant. Are you with me? Okay. Now, let's pretend that we have 4 inches of rain. And it rains and rains and rains. Good for us, we need it. But all that water is washed into this pond. There's like 2 feet of water in this pond. What else is in the pond? Endospores are there. What else is washed into that pond? Fertilizer, waste. So you have deer poo, squirrel poo, whatever, right? Yeah, whatever. You have grass clippings. You have leaves. All that is nutrients, right? In addition to that, you know, Carla ate a Taco Bell taco and dropped on the ground, and she's not like me, she's gonna pick it back up and eat it. Let it just sit, and so that's all gone in there, right? So whatever was on the land is now in that pond. So would everybody agree that there is an abundance of nutrients in that pond now, and also an abundance of things that washed in with it, right? This is the lag phase. This is the organisms getting used to the environment. There's a whole bunch of nutrients in. You have to think about enzyme systems at this point, right? What enzyme systems are at play here? Well, there are 2 types of enzyme systems. One of them is called constitutive enzymes, and the other ones are called adaptive enzymes. Constitutive enzymes are already part of the genetic makeup of whatever the microbe is in there, right? They have the ability to produce these enzymes to break down whatever might be in that pond, cellulose or whatever, okay? Good? The bacteria are really interesting because they have operons. Anybody ever heard of an operon? An operon is a set of genes that only gets turned on when the bacteria needs it, right? And so there's this lactose operon, there's a trip operon. We're not going to talk about operons in this class, but know that the bacteria have a lot of ways that they can survive.
Are those in the endospores?
They're in the genetic code of bacteria. E. coli is the big one. Okay? It's the one that's been most studied. Okay? Good? Everybody with me? The other enzyme system is adaptive. Right? Given an amount of time, if we allow the organisms to come in contact with these substrates, these nutrients, they will learn how to break them down. Plastics, styrofoam, all these things. Yep. Good? Everybody with me? So at this point, there is an abundance of nutrients. There's an abundance of microorganisms. They're all getting used, and then the growth starts. And the growth, when it starts, it goes logarithmic, right? And so when it goes logarithmic, remember, log stands for exponential growth. So in this case, the amount of growth that is occurring greatly outnumbers the cells that are dying. There's so much nutrients in the environment that for gram negs, every 20 minutes, 1 gram neg becomes 2, and for every 30 minutes, 1 gram pos becomes 2, right? It's exponential. It just goes like crazy. Are you with me? But when you have that kind of stressor on the environment, eventually what's going to happen? The nutrients are going to start becoming depleted. So that transition is to the next phase, right, where at this very pinnacle of this curve, growth is equal to death, right? And after that, nutrients become scarce, the organisms start to multiply less rapidly, right? There's not as much nutrients. And that transition is into the next phase, the rapidly declining phase. And so here, not any nutrients, cells are dying a lot more than they are growing. And what happens to the cells that are dying, Jinjin?
I'm sorry, can you repeat the question?
What happens to the cells that start to die in the D phase, in the rapidly declining phase? They are waste, they become nutrients for those that are living.
The cycle starts over.
Now, oh, she's already stealing my thunder. You are good. I like you a lot.
They start making endospores again.
Oh, wait, don't steal my thunder. Wait. I haven't finished telling the story yet, okay?
Sorry.
So these cells start to die, and eventually they come into the death phase, but I've made a big effort to make sure that my curve does not hit that x-axis. Why? Because if it hits the x-axis, the population will become extinct. And even if that pond dries up again, is there life in that pond? We just talked about it, right? Those that can survive minimally, those that go dormant, all that happens, and it's cyclic. Okay? Life is cyclic. We're born, we grow up, we get in trouble, we get wiser, we have kids. They do the same thing. They grow up, they get in trouble, it's cyclic. Everything is cyclic in nature. Are you with me? So what would happen, Miss Samantha, if we had a rainstorm happen right here?
The things that were dying would repopulate.
You have that whole cycle happen all over again. Okay? Now, Eric, let's answer your question. Where do endospores begin to start to form?
When they're getting stressed.
When they get stressed, and when is that on this curve?
I would say D.
D, they're stressed out already. So they're going to start to form here. Right? And by the time they're here, they're fully formed. And they go dormant here, right? Which really is here if we start all over again. Isn't that cool? This happens for everything that's on this planet. Now, it doesn't happen exactly like this for everything, because with humans, we just don't divide and form two cells or two organisms, right? We have to find somebody who produces sperm and produces ova, and then we have to produce another thing, right? Are you with me? So it's a little bit different with us.
Yeah, we're still multiplying like crazy.
Oh my God, we're really good at this. Are you with me? Yeah. So where our humans are in this curve, do you think? Okay. No, we're not dying yet. Okay.
I feel like we're at B. We grow more than, we have more
We're
of a growth than we have a
slightly,
decline.
we're probably around here somewhere, right? So it was just announced, 3 months ago, 8 billion people on the planet.
8 billion?
8
Oh my
billion.
God.
Alright.
Too many.
That's a lot.
We get a few natural disasters, we can change that curve.
Well, that's what pandemics are for.
I was just about to say that's cool.
However, this last pandemic that we've just gone through was not the killer that we were predicting.
And
they still blow it up like it is.
They still blew it up like it is.
Insane amount of people over risk.
And they are. And they were.
They died.
And some died, but we didn't lose 30% of the population of the world like we did in 1918. The Spanish flu of 1918 wiped out 30% of the population. That's what I was expecting. I'm still expecting it.
The Freon, right?
No, it's gonna be influenza. It's gonna be flu. That's the only thing that makes sense.
But we learned that we always have a vaccine for the flu.
Except the one that's out there right now is called H5N1. Look it up sometime.
It's from Australia.
I don't know where it's from.
The
It's from
vaccine-
somewhere.
The vaccine that we have for the flu does not always.
No, sure.
Okay, hold on.
Protect
That's a
against-
different story, but that's true. The vaccines that we have for flu are based on models. We're looking down at the southern hemisphere during their flu season, and we are picking what was most problematic to them, and that's what we make the flu vaccine for. But the one that I'm talking about, H5N1, we were trying to make a vaccine for it right before the pandemic hit. And then when the pandemic hit, because we thought it was going to be bad, and don't get me wrong, it was bad, but it wasn't H5N1. H5N1 has a 60% mortality rate.
Is that the same one that was killing a lot of infants right when COVID started?
I
in
think that was
2019?
H3N4. I'm not sure.
Is it the chicken one?
It's a chicken.
That's the one that you're saying is very harmful.
The bad one.
I know my pediatrician for my daughter had told us that like he wasn't scared of COVID at all, but he was terrified of this flu. They're not terrified, but he was worried about this flu that was killing.
I'm
a
not
lot
scared of
kids.
either one of those things, but I don't really want to get sick. And I've had COVID. I've had it.
Yeah,
It was uneventful for me.
Same.
Or as Jessica would say, it was unremarkable. Right? I didn't know I had it. My wife said, "You look bad." I'm like, "What?" And then she came in with her little swab. I'm like, "Oh hell, you're positive." I'm like, "Oh, fine." I didn't even know I had it. It was just normal for me. But 5 years ago, we both got flu. And we both got vaccinated. I felt bad for 2 days and then I was fine. It took her 4 weeks to get better. So everybody's physiology and everybody's immunology is a little bit different. And even if we live in the same house, we got the same influenza strain, it really affected us very differently.
I got super sick from H1N1.
Yeah. Which that was, and don't get me wrong, it was a weekly.
Yeah, but
Hey.
I was sick for like almost 2 months.
Yeah.
To go to the doctor.
So the only people, and I'm not trying to minimize what happened with H1N1. I'm not saying that the people we lost, I'm not saying that I'm not empathetic to that.
You mean
But-
COVID?
No, H1N1 was swine flu, 2009.
Oh, yeah. I was like, that does sound familiar.
You were a baby,
I know
you
it.
were like 2 years old.
I was not a baby.
Fine. But anyway,
I was like
H1N1 was swine flu, and the people that we lost, less than 1% of all total cases that got swine flu died. The people who died already had some other comorbidity. Right? I'm talking about a strain that will not be picky. It's gonna infect everybody, and everybody's gonna get it.
So what causes it to be?
So strong and nasty.
Oh, they have it in cats and dogs.
So the one H5N1 is a bat and a bird. Right? That's why it's called avian
Oh,
flu.
yay. I have a bath in my house.
I do too.
I was looking at the website for the WHO, for globally for the COVID updates, because I got on that quite early on, right, just want to tell you,
Mhm.
just because we're terrified. The 6.5 million deaths.
Worldwide.
Yeah.
Right.
600 million cases, confirmed cases.
So it was a significant
But-
pandemic, but it wasn't a killer.
That's entire
There was
nations.
also people who would die from heart attacks and they'd say it was COVID.
But if it's
That was
related to
proven.
Could
COVID,
have been
That
COVID
was
pushed
proven.
over.
but if it's
Yeah,
related to
but-
COVID, would they have
Like
had
there
a,
was some
what,
hospital that had like a big, where was it, Boston or somewhere, some big town city that, they got in trouble for it. Because they had wrote off, a bunch of deaths as COVID, and they had to redo.
But that was not the norm.
The amount.
That was not the norm. If that happened, that's sad,
Mhm.
because we're only as good as our data,
Yeah.
right?
Well, that was the
Just the
problem with
US,
their data
US
was twisted.
worldwide.
No, yeah, I get it, I get it. I'm not trying to minimize the fact that COVID was a bad thing. But in my mind, as a trained epidemiologist and microbiologist, COVID was nothing. It was not scary. I am not scared of COVID.
So you
I
think
respect
the
it.
avian flu-
I respect it. What's that?
You think the avian flu is gonna hit this time, this winter, like my baby's about to be
No,
born?
no. It's gonna happen sometime in my lifetime. I cannot predict it. It'll happen in my lifetime.
They're saying that it's already in the US.
It's here.
Yeah.
We have cases, but the only cases have been from people who are working with livestock and acquire it through the dust from working with chickens or ducks or whatever.
And you said the new flu vaccine will help,
The new flu
or
vaccine?
just the flu vaccine?
Not the bivalent COVID one, no. Not the flu, the seasonal flu
Yeah.
for this year, no.
Oh.
This has to be a special vaccine against it. And we're not doing any work with it right now because of COVID. Yeah.
I know when we got COVID in my house, I'm really big on vitamins, and I always keep like lots of extras for when we get sick. So when COVID happened, I took like zinc and oregano and stuff like that. Do you think that?
I
taking
also
those kinds
take
will
zinc
help?
and elderberry.
Mhm.
I have it in my
I don't
house
take
at all
zinc every
times.
day, but just when I'm feeling a little sick. I
So
will
both
take
of those have mechanisms that will not allow viruses to replicate,
Mhm.
and we'll talk about that when we talk about control of microorganisms on Thursday, and on to next week. Yes, sir.
Do you think that, the vaccine for COVID would like, for the H5N1, would it like kind of prevent,
Not at all.
I don't know?
Not one bit. Not even point 0.0000001%. They're so different.
Stay away from chicken farmers for now.
Just be normal. If you see somebody that looks sick, stay away from them, right? But you can't control what's gonna happen. Because there's too many people in this world that don't understand microbiology.
That's the big lesson. They'll repeat the same mistake with the deadly virus, or it's gonna be the same ramshackle response.
That's what I was trying to point out the other day when Samantha said, well, you didn't learn anything either. I'm like, yeah, you're right. Because I'm not really doing anything different.
But you also shouldn't, because the more you expose yourself to things, the more your immune system works.
When H5N1 shows up, I will isolate. I will retire. I'm ready.
You got a ranch and everything.
I've got everything I need.
Zinc and elderberry are mechanisms that prevent viruses from replicating.
Oregano does too. It's a natural
Okay.
antimicrobial.
Yep.
It's so interesting, because
But
it
you
goes
can
back
put
to
it in your body a
the
little
principle of survival of the fittest,
Maybe
right? Like, I just, it's so, it blows my mind because like, it's still an ongoing thing, and it's been, and it
It
will
will be an
forever.
ongoing
Yeah.
thing forever.
And you can't pick your genes and everything, so initially, you're gonna die or you don't.
That's true. About 10% of the population, no matter what the infectious agent is, 10% of the population statistically will never get sick from it. My wife is a 10%er for COVID. I had it in the house, she never got it. We've been exposed to lots of people.
Would you
So
still
she's never
consider,
had COVID.
She's never had COVID.
would you consider someone a 10%er if they had, like, they tested positive but they never showed symptoms?
No,
for
because
it?
then I would fall in the category. And I had it. So I was proud of the fact that I was a 10%er until July, and then I got it. And I'm like, oh, I'm out of the club. I won't be invited to the big party.
My daughter's a 10%er.
My wife is a 10%er. Now I say that, now watch, she's gonna get COVID this weekend.
Me and my husband both had it, and like my daughter sleeps with us, and she's never got it.
So
never
all
had it.
this stuff we just talked about is right here for you. I just like to talk about it on the board because it makes more sense. And then math is always important because remember, this standard growth curve can describe anything that's happening in an infection in the body, and contaminated food, in the environment anywhere, right? But mathematically, it's always important to think about how this might work, right? So here is a formula that I need to become very proficient with, right? So I don't do much math in this course, but I will do this math because it's important, right? It's always important to be able to understand how things can be extrapolated so that we understand what's going on. So NF is the final population, NI is the initial population, 2 is a constant for bacteria for binary fission, right? And little g to the exponent is the number of generations, right? So our practical is next Tuesday. Our review day is Thursday. Let's say that Amy invites, I'm a student, not a teacher.
Would you say the 2 was, I'm sorry?
The two what?
Would you say the 2 was for?
It's a constant for binary fission. 1 cell becomes 2. Okay? So Amy, I'm a student. Amy invites us all over to her house to study for the practical on Saturday. She says, everybody come over at 9 o'clock in the morning. We'll study for 3 hours and then we'll have lunch together at noon. All right? Well, Jess and she calls me up. She's up at 7. She goes, I'm ready. Because you know how Jess is. She always likes to be ready. And she calls me. Let's just go over there early. So we show up at 8 o'clock in the morning at Amy's house. She walks into the door and she's wearing her Big Bird shoes and says, come on in. And she says, I'm still getting up and getting ready. Can you all help me make tuna fish salad? Are you with me? So we start making, there's like cans of tuna. Right? Jess has put them all in there. She's very clean, you know how she is. And so she puts it in there. And then she says, mix everything together. And I start mixing stuff, but then I have to sneeze. And I have everything, and I can't go anywhere, so I'm not looking around, and all of a sudden, I sneeze in the tuna fish. There's stuff in it, I'm like, and nobody's looking, I just mix it in there. Are you with me? Nobody knows, right? Are you with me? So then, that's 8 o'clock in the morning.
Oh no.
We eat lunch at noon.
Delicious.
And nobody knows, but several of us get sick after it, and we want to know what the population of organisms was that made us sick. Let's just say, for the sake of argument, when I sneezed in the potato salad, Ms. Valencia, I inoculated listen to the words I'm using, I've inoculated the tuna fish with about 1,000 CFUs, okay? We now know everything we need in order to figure out how many cells made us sick. Good? Let's talk about this. So, what do we know? Tell me what we know.
You have 1,000 CFUs.
You have 1,000 CFUs. What else?
4 hours out.
4-hour period, right?
And the initial population.
Initial population 1,000. What's the generation for, let's say, I don't know, Staph aureus? We'll just make that one up. Number 1, food poisoning. What's the generation period for gram positive?
30?
Good. So now we know. Here we go. The final population is equal to the initial population times 2. And what's 2, Isabella? What is 2? 4? Binary fission. How many generations?
Oh, so that's 4 hours divided by 30, correct?
4 hours divided by 30 is what?
8 generation.
That's the answer. And when you do this for me on the examine poo, you can stop right here.
Okay, so you don't.
If you want to figure it out, it better be correct,
No.
because I'll check the math. By the way, does anybody know what this is? I think it's 256. So 256,000. And let's check, because we know that binary fission means doubling every generation. And we have how many generations? 8. So if we start initially with 1,000, then we have 2, 4, 8, 16, 32, 64, 128, 256,000 CFUs made somebody sick.
You're right.
Every once in a while I get it correct. Good? Isn't the math beautiful? The math always works out. Yep.
So in that little bit of tuna salad that I ate, There were 2,500
No, there was a lot more, but that's how many made you sick.
Yeah.
Okay? Questions? Will you be able to do this for me on examining food day?
Write it down, yes.
I have a video for it. You can look at it. On Blackboard. I did it.
Already?
Yeah. Well, I did it last semester or something like that. Because people are like, I don't get it. I'm like, I got it. I'll show you.
Where is it?
It's on Blackboard. Underneath the content. It says population curve or something like that. Population formula.
Is it under lectures?
Yeah.
Oh, using the population equation to solve microbiological problems.
Yeah, that's it.
What's the growth rate for, gram-negative?
20 minutes.
So 30 minutes was the gram positive?
Mm-hmm. They're slower to grow. Okay?
But we don't really know whether it's what kind of bacteria it is, right?
I'll tell you. Fair enough. The two that I always pick are either Salmonella, which is a gram-negative, or Staphylococcus. Or Staphylococcus aureus, which is gram-positive.
Are we gonna end up going over our test today?
Yes. I was waiting, Tay, because some people hadn't got here yet.
I was just wondering.
Yeah.
Are you gonna give us a heads up on, I don't know what to expect for this lab practice at all, I have no idea. Is that written down someplace or are you gonna coach us through?
Both.
I think there's,
So on Thursday, Eric, the whole lab is reviewed.
Okay.
Amy said we
Ecology.
should have a review.
Good job, Amy.
So I already put that in the schedule. Because I knew she was gonna ask. Okay? Let's talk a little bit about ecology, and it's not really ecology. It's really things that can affect the growth of microorganisms. Okay? And so if we think about this for a minute, it's important to understand that even though we're talking about this much of the total science of microbiology, microbiology is this big. All the cycles of the world, of the planet, are based on the microbes. The hydrogen cycle, the nitrogen cycle, the carbon cycle, the sulfur cycle, all those cycles are based on the microbes doing their job, right? Something dies, it's the microbes that replenish nature by breaking down those things that have died and putting the nutrients back into the environment, which then they can take up into their systems and continue to grow.
I think when we went to Louisiana not too long ago, they have these like crawfish, little like ponds or something. And I heard that they use that for initially, once all the crawfish dies, they use the nutrients of the shells for
Fertilizer.
Yeah, for fertilizer.
Good fertilizer.
They usually do it in rice bags.
Mm-hmm.
I thought it was really cool because we drove by and we at first didn't know what exactly it was, and then
You don't know what a crawdad was?
I'm from Germany. We don't really do that, so
Did
we don't really have that much space
Did you eat the
anyways.
crawdads?
Say it again.
Did you eat the crawdads? The crawfish.
Crawfish.
You're messing with me right now.
No,
I'm offended.
We call them
No,
crawdads.
I've had it once before.
Did
but
you suck the shell?
I sucked the shell.
Okay, good. You're a true Louisianian. Right?
But I thought it was really interesting because I didn't know, and until someone told me and pointed it out, it made perfect sense. But it just kind of is interesting.
I'm glad that they're doing
Or,
that
like,
and not just
being resourceful.
pile them up and let them stink.
Yeah.
You can use dead things for lots of things.
We've found like crawfish in the excess that we had left over in our compost.
Yep.
It stunk so
You'll-
bad, but it was.
You'll
worth it.
see my compost in a minute. I'll show them to you. But I have 3 composts going all the time, and everything that's produced in our yard or in our kitchen goes into the compost.
We have a really big one.
One more time.
I just put a branch of eucalyptus in the compost and makes everything smell good.
Well, that's
I want my compost stinky.
Do you have like possums and stuff?
Oh, lots of possums come over, and we have a game camera. We check them out. The possums act.
Are yours just too small to have it?
You have 3 different types of compost?
No, I have the same compost. It's the same.
Just time periods?
I'll show you. Yeah, I'll show you, man. It's a pretty good setup, I think. So if we think about the things that can affect growth, here they are, right? So oxygen, temperature, pH, osmotic pressure, nutrient concentration. I'm not gonna talk too much about nutrient concentration because we just did, right? But let's talk about the others, right? And so let's think about oxygen requirements. Now you guys, I don't have to do much with this already because we've already done it in the lab, right? We use thioglycolate. We have lots of ways we study the oxygen requirements. And just to review, what is this type of organism growing in this tube right here?
Anaerobic.
Anaerobic. It's not growing anywhere where there's oxygen. What about this one here?
Aerobic.
What about this one here? It's growing more at the top, but growing throughout the tube. What is that? Facultatively anaerobic. And what about this one here? It's growing slightly below the meniscus, about halfway down the tube, but not at the bottom of the tube. What is that? Microaerophile. See how much you know? Here you know it. It's beautiful. We can put it into theory now, right? Because you all know it, right? And an organism that prefers CO2 versus any other type of gas is called a capnophile. Mhm. Good?
Oh, it's right there.
Capnophile. Next slide.
I have it written down.
Yeah.
Good? Temperature is also important. So we think about this, we classify organisms based on where they'll grow. Saprophiles will grow in cooler temperatures. So I always like to say freezer or refrigerator. And what most people think is if you put stuff in the refrigerator, the freezer, you're going to kill them. That is not true. They will continue to grow. They'll just grow slower. That's why milk or any other food can spoil in the refrigerator. And that's why other things can spoil in the freezer if it's there long enough. Okay? So what I think, instead of you learning the range, I think if I asked, what type of organism would grow in a refrigerator, you'd say a saprophyte, right? Good, yep.
So a lot of the molds and things like that grow in fruits, they're stocked. So I keep all of my fruits in like big mason jars. So they're stocked by that lack of oxygen.
That is correct. Molds are completely aerobic.
Okay.
If you take their oxygen away, they will not grow. And the interesting thing about molds,
Not apples. I put like strawberries and cherries. I've had some cherries for like 3 months now, and I still eat them.
Yeah.
And they've been in a mason jar, a big mason jar.
but the thing about
They're still fresh.
the cherries you have to be careful with, if they have yeast, the yeast will grow anaerobically, and you'll get alcohol, which might be.
Yeah, I don't mind it. Just don't give them to my daughter.
So, mesophiles will grow at middle temperatures, and that includes room temperature and body temperature. Okay? And thermophiles will grow in temperatures that are slightly above what we can reach. Or actually, they're above what we can reach, right? Thermophiles are never going to grow in us. Yep, yep.
So the type of bacteria that grow on like fresh chicken eggs, why, like, so they say that you can leave them on the counter for a long time, like if we have a jar
Yeah.
or a basket of chicken eggs that we've had for like a month and they're still fine.
Yeah.
Why does nothing grow on it?
So there will be things that are on the eggshell, but as long as there's no cracks or breaches in the egg, they're never going to
Then
get on the
inside
inside.
is protected.
They're never going to get inside.
So why do we refrigerate eggs?
Because we're Americans.
Because you're told to do it and you just do it.
That's
If you go
it's
to
American
other
culture.
countries, they
Yeah, I
don't, even
know.
at the grocery store, they don't even have the eggs in refrigerators.
It's just American.
I read that it's like the way they process the eggs and stuff.
Not all eggs are processed. Some of them are actually dipped in a 10% bleach solution, but not all of them.
I was just curious because eggs are porous, that's why they tell you not to.
They
let
can
them
be
soak.
porous, but once the shell is hardened, you're not going to get through that eggshell.
Okay, so like the bacteria won't go through the
No, I mean,
eggshell
so
once
just
it's-
think about when you colored Easter eggs.
True,
As long as you
inside
didn't have
isn't-
any kind of cracks or breaches
I
in
didn't
that
know that.
egg, the dye didn't get through
Mm-hmm.
into the egg. It's only when there was a crack or a breach, right? Because they're porous, but once they've hardened, you're not going to get through it.
This may seem dumb, but how come like the natural grown eggs they're like brown for like one?
That's genetics. That's just the type of chicken that produces
Like,
them.
I have red hair,
Yeah.
somebody has blonde hair, it's just like the general.
Yeah. So some of them are
What color
greenish,
egg
some of them are
the
deeper
chicken?
blue colors, some of them are brownish. Some of them are orangish, yellowish. There's all kinds of different kinds of eggs that are produced by chickens. The normal chicken that is domestically commercialized produces a white eggshell.
But if you buy eggs that are refrigerated, you need to put them in the refrigerator after, right?
The other day.
No.
Yeah, the meat and.
And that's one of my favorite foods, is eggs.
You just leave them out on the counter.
And even cooked, I leave them out on the counter. It drives my wife crazy. Why did you leave these out in?
An egg?
Yeah. Why did you leave? I'm gonna eat them later. She said, well, why don't you put it? Because then they'll get all moist and soggy, and I don't want my moist and soggy eggs. Right? So I want my, and the same thing with hamburger. If I ever make a hamburger or something, just leave it on the counter.
Same.
Why did you leave it out? Well, because I'm gonna eat it later.
My husband does not give a crap about anything. He'll just eat it like,
I don't want soggy bread.
or fridge cold.
Yeah, okay. So psychrophiles will grow in the frozen tundra, right? So I think I told you that my first master's degree, I worked with a guy named George Meyer, and Professor Meyer had done a lot of work in Antarctica. And so if you ever look at the map of Antarctica, you'll see the Meyer Desert that was named after him because of all the work
I
he's
was
done.
wondering why that's not up
So they'll grow in frozen tundra. Mesophiles will grow at middle temperatures, room temperature, body temperature, right? And here, if we take a look at Staph aureus, which is an organism that has evolved with us, it is accustomed to living with us. If you put it at 22, it'll grow, but it's not happy. You put it at 30, it starts to get happy. If you put it at body temperature, it throws a party because it's happy. Are you with me? And that's the interesting thing about organisms. They grow optimally at some temperature, but not at other temperatures. Okay?
I grow better at hot temperatures.
Yeah. So here are the thermophiles. Thermophiles will grow where you think they cannot grow, right? And the volcanic ponds, right? Hot, very hot. And even in the geothermal vents down at the bottom of the oceans, right? The archaea have been proven to grow down there. It is an amazing thing, right? I can remember several years ago, the Discovery Channel had these 2 episodes of the research that had been done in the geothermal vents. I got so excited. I made popcorn. I was just watching the show, and the dog was with me. And he would get popcorn too. It was such a great show.
So like that's all that, when I went to Yellowstone, that's
So those are either
Okay.
thiols. They're bacteria that like the sulfur or the heat. The archaea. Okay, good.
I'm lost for a second, sorry. I was trying to go down the slides and take notes on what you were saying about the different ones, like in different environments. There's one that is like our body temp. Where is that
Mesophiles.
one? Oh, okay, sorry.
Okay.
Thanks.
Good.
Yeah.
Now, based on pH, right, organisms will grow in different pHs, right? Bacteria like kind of a middle of the range pH, right? So if you look at bacteria, right, they grow mostly between 6 and 8. But the fungi, the molds, the reason, Tay, they grow on your citrus is because they like acidic conditions. You're never going to find mold growing on soap, ever. They don't like alkaline conditions at all, right? So just think about that for a moment. If you wanted to get rid of mold, 2 things you could do is take away their oxygen or use some type of basic cleaner, and it'll kill them, right? But you can't use acetic acid because they'll just go, thank you very much for bathing me in what I like the most, acid. Yes, sir.
So is that why like alkaline water has become more popular?
No, alkaline water is a marketing ploy to get you to spend a lot of money for no good reason.
I was just wondering if maybe that's,
So what
that's a marketing tactic of
the
it.
hypothesis, the marketing of alkaline water is change the pH of your body, make it more alkaline, and you won't get any cancer. That is correct. You also would not survive. Because if your body became more alkaline, your body would shut down completely and die. The body can only survive at a very narrow pH range. And the buffering system in your blood and in your body will not allow for any changes that are outside of that range, or you will die. So if you're falling for drink this alkaline water, you're just wasting money. It's just a marketing ploy. Okay?
I think there's a History 101 on Netflix about bottled water and how much of like a scam it is. Like they're typically just tap water. I mean, they are filtered and they
Well,
do have
the
some.
other thing about bottled water, and I don't wanna get y'all excited about this, but the plasticizers in bottled water will shut down your endocrine system. So you need to be real careful about that. And don't get me wrong, you know that I use plastic bottles, but, you know, I don't really wanna add a lot of trash to the environment. So I buy one bottle and I'll use this bottle forever. I used to use a styrofoam cup, but it grossed people out because there'd be all kinds of colors and things growing in it. So I just stopped to do that. I have a really nice metal cup, but it spills everywhere I go,
Yeah,
so
they're hard to-
it's just not, it's not sound for me because I move around too much. I move from campus to campus all the time, and by the time I come from South Austin to here, I have half a cup of tea, and it's just not fun. to
So you
lose
would
everything.
say, like, any water out of tap would probably be healthier than bottled water? Cause I only drink,
I don't know that I would go that far.
I have a mold
but I
question.
only drink well water.
Well water's great, but it may not be around for a lot longer because everybody's tapping into the wells. Yes,
So what is the pH of bleach?
about 10 or somewhere around there.
Okay, so there's a mold problem at my dad's place, black mold. He
Where?
sprayed it with bleach,
Yeah.
and I heard or read that can make it so that it's like almost like a powder form, and then you breathe it into your lungs.
That is true. So instead of spraying it, wipe it.
Okay.
So take the bleach in a towel and put it on the mold. And
Would
then
ammonia be better?
no, bleach is a lot better. Bleach not only is alkaline, but it also has a contact time of about 3 seconds. And because it's a halogen, it will kill the mold in about 3 seconds. So bleach is one of the better of all disinfectants.
Okay.
And you only need a 10% solution. The other thing that would be just as good would be hydrogen peroxide. It has a lesser contact time, about 0.8 seconds, and it will kill just about everything it comes in contact with. But again, you want to wipe it, you don't want to vaporize it, because you could actually put the spores of the mold into the air. That's not necessarily going to be bad for most people, but there's about less than 1% of the population is highly allergic to the molds that can be growing on sheetrock or whatever.
Okay.
Okay? Questions? All right. So, ever see bacteria grow in coffee? No, you haven't.
What about the mold on the top?
That's
of
not
the bottle?
bacteria. That's mold. That's a fungus. It lacks acidic conditions.
Oh,
Ever
yeah.
see bacteria grow in orange juice? No. Ever have orange juice that's bubbly and you taste it's more alcoholic? Yes, sometimes I let it go. I'm like, okay, ooh, good. That's the yeast fermenting.
The mimosa for free.
But those are both molds. They prefer acidic conditions. Right? Bacteria will not grow in coffee. It will not grow in Coca-Cola. It will not grow in orange juice.
The things
Because
that
it's
it can
too
do.
acidic. Yep.
So an apple juice,
A what?
an apple juice,
Yeah.
would that be the same thing?
Apple juice is not as acidic. It's more sugary. So I think bacteria would grow in apple juice. I don't know that for a fact. I never really thought about it. Okay. Yep.
I forgot to ask you, I have a water bottle that my fiance left, and there's stuff growing in it. Do you wanna look at it if I?
Yeah, bring it. Bring it out. We'll plate it. See what's going on. I might be telling you before we plate it.
You're probably gonna sip it up.
All right. So if you take a true acidophile like Euglena mutabilis, it grows between 0 and 1 pH. That is really acidic. And you just put it in normal water, it will die instantaneously. It could not handle the pH change. Okay? If you want to be a hero at your house, use an alkaline cleaner 1 week, 10% bleach. And the next week, use an acidic cleaner, acidify your bleach. Add a little capful of acetic acid, vinegar, to your bleach, you change the pH, and the organisms can't handle the change in pH and they die quicker.
So you mix vinegar with bleach?
Do you add any water or?
Well, so 10% bleach with a little cap full of vinegar.
What type of vinegar?
What kind of what?
There's so many
White
types.
vinegar.
Oh, white vinegar. Don't get the expensive kind. No. Don't buy Mother Hubbard's vinegar. Don't do that. Just get the
From the mother.
the cheap stuff.
No, he said vinegar from the mother. He said Mother Hubbard.
Okay, osmotic pressure. I expect that you already know what isotonic, hypertonic, and hypotonic are. Isotonic, if it's in an isotonic environment, Tay, what happens to the cell?
Isotonic?
Yeah,
Stay the same.
nothing. It's happy. What happens in a hypertonic environment, Tram?
Hypertonic, there's too much concentration inside the cell, so it will.
No, outside the cell.
Outside the cell.
So what happens to the cell?
The water will leave the cell.
And so what happens to the cell?
It shrinks.
It shrinks, very good. And a hypotonic environment, what happens, Ginjin?
Bursting.
It can burst, right, because water rushes in. And if the cell doesn't have any way to deal with osmosis, it's gonna burst. Are you with me? Good.
Cell
But-
lysis?
Mm-hmm. Plasmolysis, yeah. So the other thing I want you to know about that is that there are some organisms that are halophilic. How many people have ever heard of that? Halophilic.
They love salt.
They love salt. Halophilic can tolerate to about 10%. Okay. Staphylococcus aureus is the #1 halophile in the entire clinical world that we worry about.
What is it,
Staphylococcus aureus.
That's aureus.
Uh-huh, S. aureus. What is the #1 cause of food poisoning? Staphylococcus aureus. Most people don't know that. #1 cause of food poisoning.
Right off our skin into the food or whatever.
Here. This is where it normally is. It can be on the skin also, but it's mostly here.
Inside the nose. or-
Inside the nose.
Inside the nose.
Yep. Good. What is the number one holiday for food poisoning? That is correct. Next, second one.
Thanksgiving.
Thanks, and the third one.
Christmas.
And then after that, we don't care. Birthday party could be it too. Yeah. Good? All right. So the last thing we talk about today is symbiotic relationships. It's really all about nutrients, right? These relationships can be obligatory, they can be non-obligatory, but they all center around relationships. And we call this relationship a symbiosis. It is the relationship between organisms, and it's all about nutrients, right? So the first type of relationship that we have is called mutualism. And in mutualism, what you have is you have the relationship between organisms, and all of the organisms involved in the relationship mutually benefit from the relationship. This is my favorite example. These are lichens, L-I-C-H-E-N, not that kind of lichen, L-I-C-H-E-N. And this is a combination of 2 molds, 2 fungi, and 1 alga. The algae produces sugar photosynthetically, and all the organisms use that sugar to grow. And the mold protect the algae from being destroyed, from being desiccated, from all that stuff. It's a pretty good relationship. All of the organisms mutually benefit from the relationship. Okay? Commensalism is the relationship between organisms where one of them benefits from the relationship, but the other one is neither harmed nor benefited, right? So here we have examples of a whale and some barnacle. The whale does not care that the barnacle is growing on its fin, but the barnacle has a place to live. Yeah? Here's a bromeliad. These are beautiful tropical flowers that in Puerto Rico and Hawaii and places like that. We also have them here. We call them ball moss here, right? The ball moss grow, but they're epiphytes on the tree. What does that mean? They're an epiphyte. That means they grow on the surface of the tree, but they do not take nutrients from the tree. So the ball moss, the epiphyte, has a place to live, but the tree is not being harmed. That is different than mistletoe. Mistletoe is a parasite. It sticks its vascular structures inside the tree, and it's taking nutrients from that tree all the time. Do you know why we kiss each other under the mistletoe at Christmas?
No.
Because we didn't know what else to do with it. It was bad and we want to get rid of it. We came up with the idea of making it a kissy place for Christmas to try to kill the mistletoe. If you see mistletoe growing out of your tree, you should cut it out. It is destroying the tree. Good. You learn something new every day.
They pulled mistletoe off their Christmas trees every time?
No, off of any tree.
Does it tend to grow here though?
Oh God, yes. You want me to bring you some in?
If you want to.
I'll bring you some in as we
When,
get.
when does it grow? Like
It's
all year
getting
round?
close to forming berries now.
Oh.
So I will pull it out of. It grows really well in my.
I
in-
probably have it at home.
in my
but I don't-
hackberries. It
I
grows
don't really
really
know
well.
what it looks like.
I'll bring you some. There's all over. We'll probably go out there and walk around a little bit and find them.
I mean, I do have these things that grow on certain trees on the property that have berries, but I don't know if that's, there's so
Be
many.
careful, it might be poison.
I don't touch them.
Okay.
Or what are the little lumps?
I don't
They
mess with
look like
plants.
little woven ball, like branches. They're all over the tree.
Those are insect galls. Take a picture of it, make sure, because I don't
Yeah.
want to tell you incorrectly.
Yeah.
Okay?
So it almost looks like it's like a parrot. It almost looks like it's destroying the tree, but I don't know if it is.
Take a picture.
Yeah.
And let me see it. Okay? And then you have parasitism, right? Give me an example of a parasite. Do not say my ex-boyfriend. Roundworm. Roundworm. So here's a tick, right? There's a roundworm. So look at the tick. The tick is embedded in that tissue. It's taking a blood meal. It's benefiting the relationship. But the other thing that's in that relationship is being harmed. In some cases, harmed and not even knowing it, because some of these ticks carry things like typhus and Lyme disease and all kinds of different things, right? So if they get on us, we have to be leery about that. Yes,
What's the one in the wasp?
and the what?
In the wasp, I think there was some that go into the, like I don't know, a certain type of wasp.
Oh, I know what you're talking about.
I have
And
not.
they use them as a host, and they like lay their, are you talking about the one that like lays
Yeah.
eggs? I know what
They
you're
die
talking
at the end of the
I'm
wasp.
not too sure about that.
Yeah, the egg from the parasite grows and it eventually kills the wasp and
Close.
eats it.
So back when the whole pandemic was still a thing, there's like a trend on TikTok where like we're saying, oh, eat papaya seeds because like it kills all the parasites in you and like you poop
When
out
they say you
more.
can see it come out. or
Is that
something.
true?
No.
It would be eating them.
Yes,
So I don't understand. I thought that symbiosis meant that like it was neither like help nor foul.
no, that's commensalism. Symbiosis just means there's a relationship between organisms.
They're just hanging out.
Or hurting each other or helping each other. So
It
under
could go either way with symbiosis.
so think of symbiosis, symbiotic relationship as an umbrella, and underneath that you have mutualism, commensalism, and parasitism as examples.
Would a flea be considered a parasite?
It doesn't really fit the entire.
Because like if a
It
dog
is.
is
It's a
covered in
transient.
ticks,
It's a transient organism.
it's just kind of there.
So in order to be a parasite, you have to live with the host.
Uh-huh.
Fleas a lot
They
of times.
kinda do, but like a tick, like if an animal's covered in ticks, they could die, but if an animal's covered in fleas,
They could
don't.
still die. But
If there's some kind of, okay.
So symbiosis just means relationship. It doesn't define the relationship.
doesn't.
It just means.
It
that
doesn't
they're
describe the
connected.
relationship. It just says it's a relationship. Yes.
Would humans be parasites on Earth?
No.
Are you sure?
We're an invasive species.
That's not a parasite?
Mm-mm.
Even though we're destroying everything? I have
We're an
had it.
invasive species. We don't have to be, but we choose to be.
Yeah.
Yep.
I had a teacher and it was a very extreme correlation, but she was like, technically your babies can be parasites while they're growing. And I was like, this is just so bizarre to me. Like, it was just such an extreme thing, but
The baby's not living. It's not living off of you. It's
Yeah,
living
living
with you.
yeah.
You're nourishing it. That's
Yeah.
different.
and it gives you something.
True.
I feel like you could make it become kind of parasitic if you don't take care of yourself.
Yeah.
Because the baby would get, but it's not really.
Yeah, let's get away from babies and parasites. Okay, it's not gonna do it.
When I was doing the morphology thing on Klebsiella, I was getting the impression, I didn't know what commensalism was, so I was trying to figure out.
So Klebsiella is considered to be an opportunistic pathogen. It is mostly commensalistic, but if it gets into a body part of the body where it doesn't belong, any place outside of the intestinal tract, it could cause infection.
Right. Well, one of the things that I saw was that they said that it, in a study, that it produced a film around it that hindered other Klebsiella species. Right? And so I got the impression that commensalism, and I was looking for simple definitions on it.
You
Right?
could have called me.
Yeah.
I would have told you the answer.
Okay, you're telling me. Yeah, that I thought that commensalism, they're saying, hey, it exerts some sort of inhibitory effect against other bacteria.
No, that is antagonism.
Okay.
That's this.
Okay.
Antagonism is when there's an organism that is releasing molecules that inhibits other organisms from getting close to it. So here is a bunch of fungi growing around this bacillus. The bacillus is producing a very powerful antifungal called amphotericin B. The B stands for bacillus, okay? So this is a very powerful antifungal that's not allowing that fungus to get near it. The fungi do the same thing. They produce molecules that do not allow bacteria to get near them. What do we call those things? Antibiotics.
Yeah.
Oh.
Good?
So satellitism is a branch under commensalism.
Satellitism is essentially when you're growing, when an organism is growing on some medium, and it's producing nutrients that other organisms need, and those organisms grow near them. That's what's called a satellitism. Okay? Good. I don't like to use synergy. A lot of people use synergy to talk about microorganisms. I do not like to use them. I would prefer to use synergistic effect or synergy when we talk about drugs. And I will when we talk about antibiotics on Thursday. Okay? The last thing we're going to talk about is the normal flora or the microbiome of the body. So these are the organisms that are on you and in you, and you should be cultivating. I think I told you that every day I eat a little bit of yogurt and mixed fermented fruits and things like that. I mix it all together with nuts and I eat that. Plus I also take a probiotic, right? So I never have intestinal problems, ever. That's probably too much information, huh, Tran? But, you know, it just goes to showing you that if you take care of yourself, a lot of people believe, including me, that if you have a healthy intestinal flora, organisms, that you're not going to have a lot of the problems that are really causing a lot of mental illnesses to a lot of the population.
Hey, mental illness. That's what any integrative specialist will tell you. They're doctors and they have a natural mindset and they go down that rabbit hole and they prove it all. It's remarkable.
I've gone down the rabbit hole.
Dr. Mark Hyman